Clarity Clinical Solutions.
What if a new drug is not better than the old one, but still good enough to use? Is that a failure or a success? In clinical research, there is a whole class of trials designed to answer exactly this question. They are called non-inferiority trials, and they help us decide when a treatment that is not the best is still worth using. A non-inferiority trial is designed to show that a new treatment is not worse than an existing one by more than a small, pre-specified amount. Unlike superiority trials, which aim to prove the new treatment is better, non-inferiority trials ask a more modest question: is the new treatment at least as good as the old one within an acceptable margin? This kind of trial is crucial when the new treatment offers some other advantage, like fewer side effects, lower cost, or easier administration. There are three types of comparative trials. Superiority trials try to show that treatment A is better than treatment B. Non-inferiority trials try to show that treatment A is not worse than treatment B by more than a small margin. And equivalence trials try to show that treatment A and treatment B are essentially the same, within a margin on both sides. The key difference is the margin. Non-inferiority only looks at one side: is the new drug not too much worse than the old one? Equivalence looks at both sides: it must not be too much worse or too much better. The non-inferiority margin, often called delta, is the maximum amount of effectiveness the new treatment is allowed to lose compared to the old one. Researchers choose delta before the trial starts, and it must be small enough that losing that much effectiveness is still clinically acceptable. For example, if the old drug cures 90% of patients, and the new drug has fewer side effects, maybe we accept a margin of 10%, meaning the new drug must cure at least 80% to be considered non-inferior. Choosing the non-inferiority margin is one of the most debated aspects of these trials. The margin must preserve a meaningful portion of the old drug's effect over placebo. Statisticians look at historical trials of the active control to establish its effect size. Then they choose a margin that retains at least half of that effect. This is called the M1 and M2 approach from ICH-E10 guidelines. M1 is the entire effect of the active control over placebo. M2 is the smaller margin used in the trial that preserves most of that effect. Non-inferiority is established by looking at the confidence interval of the difference between treatments. If the entire confidence interval lies above the non-inferiority margin, then non-inferiority is declared. For example, if the confidence interval for the difference is between -2% and +5%, and the margin is -10%, the new drug is non-inferior. But if the confidence interval crosses below -10%, the trial fails to show non-inferiority. The position of the confidence interval is what matters, not just the point estimate. Non-inferiority trials are used when a placebo-controlled trial would be unethical. If an effective treatment already exists, you cannot give patients placebo. So you compare the new drug against the active standard of care. These trials are also used when the new treatment offers meaningful advantages over the old one, like a better safety profile, a simpler dosing schedule, or lower cost. The goal is to show that the new treatment preserves enough of the old drug's benefit while offering these other advantages. Non-inferiority trials face several challenges. First, assay sensitivity. Can the trial actually detect a difference between treatments if one exists? Without a placebo group, you might not know if both drugs failed. Second, the choice of margin is sometimes criticized as being too wide or too narrow. Third, these trials often need larger sample sizes than superiority trials because the margin is small. Fourth, there is a risk of bio-creep. Each generation of drugs becomes slightly less effective, and over time, the standard of care can deteriorate. Regulatory agencies are cautious with non-inferiority trials. The FDA and EMA require the margin to be justified with historical evidence. They also require proof of assay sensitivity, meaning the trial must be able to distinguish an effective treatment from an ineffective one. The ICH-E10 guideline is the key regulatory document. It defines when non-inferiority trials are appropriate and how the margin should be chosen. Regulators are especially concerned about bio-creep, where successive non-inferiority trials gradually erode the effectiveness of the standard of care. Non-inferiority trials often require larger sample sizes than superiority trials because the margin is smaller than the effect size you would target in a superiority trial. The sample size calculation depends on the chosen margin, the expected event rate in the control group, and the desired statistical power. A common mistake is underestimating the required sample size and ending up with a trial that is too small to demonstrate non-inferiority, which is not the same as proving equivalence. An underpowered trial just fails to show anything. Interpreting non-inferiority results requires careful attention to the confidence interval. If the confidence interval lies entirely above the margin, non-inferiority is demonstrated. If it crosses the margin, non-inferiority is not established. This does not mean the new drug is inferior, just that the trial did not prove it is not worse. If the entire confidence interval is above 0, the new drug might even be superior, but only if the trial was designed to test superiority as well. A non-inferiority trial cannot claim superiority unless that was pre-specified. There are several common misconceptions about non-inferiority trials. First, non-inferiority is not the same as equivalence. Equivalence requires the confidence interval to lie within a margin on both sides. Second, failing to show non-inferiority does not mean the drug is inferior, it means the evidence is not strong enough. Third, a non-inferiority trial that shows the new drug is slightly better still does not prove superiority unless that comparison was pre-specified. And fourth, a wide confidence interval is not evidence of non-inferiority, it just means the result is imprecise. 3 best practices for non-inferiority trials. First, pre-specify everything, the margin, the analysis method, and the criteria for non-inferiority must be defined in the protocol before the trial starts. Second, ensure assay sensitivity by including a placebo group or using a sensitive patient population and endpoints that have been validated in previous trials. Third, be transparent about the margin and its justification. Regulators and readers should understand why that particular delta was chosen and what it means for clinical decision-making. So, what is a non-inferiority trial? It is a study designed to show that a new treatment is not worse than an existing one by more than an acceptable margin. These trials are essential when placebo controls are unethical, and when new treatments offer advantages in safety, convenience, or cost. The key is the margin, choosing it carefully, justifying it with evidence, and interpreting the confidence interval correctly. Non-inferiority is not about settling for less. It is about knowing when good enough is truly good enough.